Hello class and good day. This is our lecture for our module 6, namely amino acids, peptides, and proteins. Guys, I assume na this one here, this lecture was discussed thoroughly by your lecture instructor. Sir, bakit niyo po siya dinidiscuss, sir? Kasi nga, this is only an essential for our laboratory for you to understand better kung ano ba nangyayari sa mga experiment natin. And so guys, bear with me to my fast-paced discussion kung nabibilisan kayo. And feeling ko naman is masasabayan niyo ako kasi na-discuss na sa my lecture dito. Okay, simulan na natin. So what are the topic outline of our discussion here? We will talk about the amino acids. Then the proteins, which are the polymers of amino acids. We will talk about the levels of protein structures, the primary, secondary, tertiary, and quaternary of primary structures and sample of proteins. And we will skip to the part the protein verification and characterization techniques. Pero magkakaroon tayo dito guys ng ating magkakaroon tayo ng sequencing of amino acids. So simulan na natin. Okay, what is amino acids guys? Amino acids is yan yung natin. It is yan yung nagme-make up sa mga proteins natin. And we are familiar that the proteins are made up of amino acids. And it has a structure features just like this. It has a amino group and carboxyl group. And side chain na kung saan nagkakaroon siya ng 20 types of amino acids natin. Okay, mga amino acids natin, the side chains. And then the amino acids present in nature are in the form of L stereoisomers. When you say L stereoisomers as a form of this, by which mauna yung amino group natin before the carboxyl group. Kaya meron tayong term kung narinig nila. NCC sa mga peptide bonds natin. And like the carbohydrates, yung carbohydrates kasi natin are in D stereoisomers in nature. So ano ba yung mga amino acids natin? Guys, there are 20 amino acids classified by the presence of their R groups, which are dito sa may side chain natin. Okay, we have non-polar allopathic R group, aromatic R group, polar uncharged R groups, positively charged or basic R groups, negatively charged acidic R groups. And we have this. For the non-polar allopathic R groups, they tend to cluster within the proteins within a means of hydrophobic interaction. Sir, bakit po sila hydrophobic interaction? Hydrophobic means ayaw nila sa water. Hindi sila nakikipag-interact with water. Kasi nga, due to the presence of CH bonding. And we all know that this one here, the CH group, is a has a hydrophobic property na discuss sa may organic chemistry ninyo. Kasi nga, ito ay mga non-polar. Kasi sa may water natin, that is polar. And here are their R group in their side chains. For their 1-letter and 3-letter symbols, i-screenshot lang ito guys. Itong ating PDF na nandito. Here are the 1-letter and 3-letter symbol of this. So ito yung mga glycine, alanine, proline, valine, leucine, and iline natin. Sir, bakit po ba ganyan? Kasi nga, madalas sa mga kung magbabasa ng mga books natin and sa mga articles natin, the amino acids are represented by 3 or 1-letter symbols. So ito yung mga natin. Screenshot nyo lang sa guys, okay? Punta na tayo sa ating next discussion in how it's ours, guys. Sorry. Next natin is the positively charged R group. Sir, bakit po sila positively charged R group? Kasi nga, they are positively charged at physiological pH and they tend to interact with water due to their hydrophilic R groups. Sir, bakit po sila hydrophilic R groups? Kasi nga, meron silang mga NH group dito that tends to form a hydrogen bond with water. And those are lysine, arginine, and histidine. For their 1-letter and 3-letter symbol, guys, we have this, the lysine, the KHR, this KSR. So take screenshot, guys, in order to familiarize the 3-letter and 1-letter symbol of these amino acids. And then next, guys, sa ating discussion natin, we have the negatively charged and basic R group. We have the aspartate and glutamate. Sir, bakit po ba sila negatively charged R group? Kasi nga negatively charged sila at pH 7. Tapos meron silang presence na ito. And they have the net charge of negative here, which is negative 2. Which, ito lang, dalawa lang yun, the aspartate and glutamate. For their 1-letter and 3-letter symbol, guys, is ito yung kanilang one. We have the DE, aspartate D, glutamate D. Okay, next naman natin is the aromatic R groups. Sir, bakit po sila tinawag na aromatic R groups? They are aromatic R groups due to the presence of the aromatic ring or benzene ring in the amino acids natin here. And they tend to absorb at a wavelength of 280 nanometer. But sila nag-absorb kasi nga, the aromatic ring here has a conjugated property that absorbs at the wavelength, absorbs a light, either visible light here. So we have phenylalanine, tyrosine, and tryptophan. Take note guys ito yung mga natin, yung mga structures natin. And you are not required to memorize all of those. Kasi nga, open book naman tayo lagi. So, ang papadrawingan ko ay ng peptide bonds. So, always consult the structure. Okay, for their properties, for their three-letter and one-letter symbol, we have this FYW, here. So take screenshot, guys, in order for that. Next naman, guys, is we have the polar uncharged R group. They are more soluble in water than non-polar amino acids. Sir, bakit po sila soluble in water? Because they tend to form a hydrogen bond with water due to the presence of the OH group in serine, OH group in threonine, and so on and so forth, and NH group in asparagine and glutamine, and then cysteine natin. And then, ayan. For their one-letter and three-letter symbol, we have this, the S, T, C, and Q. Those are the natin. And then, itong table na to, guys, is their pKa values na makikita ninyo sa may mga titration curve natin in the isoelectric point. When we say isoelectric point, it is the net charge, has a net charge of our proteins in zero natin. And these are the uncommon amino acids that also have important functions such as the hydroxyproline, which participates the collagen formation to prevent the scurvy in our body kapag napag-intake tayo ng ascorbic acid, if you read the article sa may mga natin, mga sailors natin. We also have 6N-methylserine, lysine, and then gamma-carboxyglutamate. And then, ayan, nagkaan lang din kasi dito, guys, due to the presence of the foreign group, to the foreign elements here. Okay, and dito sa may amino acids natin, guys, we have 10 essential amino acids. When I say essential amino acids, ito yung mga hindi nasisynthesize naturally ng katawan natin. And kailangan natin silang i-intake sa ating diet. Okay? And what are those, sir? Ano ba yung sinasabi nyo mga essential amino acids po na yon? Those are the, natin, yun ay ang mga, we have histidine, lysine, threonine, we have methionine, we have the F, the phenylalanine, valine, what's this? Tryptophan. Okay, ilan na to? 7. And then, we have isoleucine, lysine, and then arginine. Para mas maano nyo, di ba? Oh, sir, hindi namin kaya memorize mga ito. Ito yung tinuro ng teacher ko para ma-memorize ng mnemonics. Here lies the most famous versatile woman I know. Right? So this is the mnemonics in order to memorize the 10 essential amino acids. Amino acids. Yun, mahalata na naman, the first letter stands for the one-letter symbol of our amino acid. So here lies the most famous versatile woman. I know, right? Yan yung mga 10 essentials amino acids natin. Next naman natin, as sinabi ko kanina, that our amino acids has a amphoteric property just like water. When you say amphoteric property, guys, it can act as an acid or base. Kasi nga, and when you add this here, it is a self-explanatory PowerPoint. When you add a base, magkaroon siya ng ito dito, magkaroon siya ng positively charge. And then kapag bitter ion, which has a neutral pH or neutral charge, ayan o, positive, negative is equals to 0. And then kapag nag-add ka ng base, it has a negative charge. But my point here is kapag nag-add ka ng acid, ang net charge niya is positive 1. Kapag base naman is negatively charged. And these are the characteristics of the titration curve for our glycine. And pinakita ko naman yung mga pKa values nila na kung saan yun yung mga pKa values nila at a certain pH there. And then these are their isoelectric point na kung saan kunin mo lang yung sum nito, yung average ng pKa1 and pKa2, and you get the isoelectric point niya. Okay? Those are the amino acids. And then when we combine the amino acids here, mag-generate siya ng H2O. Kasi nga, pag H2O. Kung sa may amino acids natin is, sa may carbohydrates natin is glycosidic linkage, sa nucleic acid naman natin is phosphodiester bonding. Dito naman sa ating amino acids, guys, ang tawag doon sa may bond na nagpa-bond between them is called a peptide bond. Peptide bond here, and it tends to form between the C and the N of our peptides and proteins for the C and N, carbonyl and amino group. Okay, so ano ba yan? Kasi nga, di ba, when you combine all of the amino acids na kahit napakadami, it will form a protein. Pero ano ba yung mga biologically active peptides natin on polypeptides? occurs in a vast range of sizes and composition. Ano ba yung kanilang mga uses natin and what are those classifications? So our proteins are classified based on their chemical composition, structure, functions, and solubility. Kasi actually, to be honest lang guys, nung high school ako nung bata, alloy proteins lang natin is matatagpuan lang siya sa may muscle natin. Sa may muscle lang. But then, as I go on to study to my pursue my degree, na-realize ko na yung palang proteins natin guys is marami palang functions na nakaka-affect to the overall or holistically our overall function ng ating katawan natin. Okay? So ano na ba yung classification ng proteins natin? The proteins are classified based on their chemical composition, structure, functions, and solubility. So it come up to my realization na ayun pala. So we have sasabihin ko naman mamaya. For the classification proteins based on the chemical position, we have simple proteins and conjugated proteins. When I say simple proteins, it is also known as homoproteins because they are only made up of amino acids. Amino acids lang yun. And those are the plasma albumin na makikita sa mga eggs natin as a storage protein, collagen sa balat natin, and keratin na makikita naman sa may buhok natin. So yung proteins pala guys, is makikita din pala sa skin natin, sa buhok natin, tsaka sa may itlog na puti. Akala ko sa may muscle lang siya. So ganun. And then we also have a conjugated proteins. When we say conjugated protein, it's a heteroproteins guys because they contain a non-protein portions. Ano ba yung mga non-protein portions na yan? Those are the other biomolecules such as nucleic acids, lipids, and carbohydrates natin. So if carbohydrates and proteins, lipoproteins, and chromosomes, nucleic acids, so chromoproteins, nucleic acids and proteins, and then lipids and proteins, lipoproteins, and phosphoproteins. So conjugated proteins and simple proteins. And then next, for the classification of proteins, we have based on shape, and there are two. There are two. We have fibrous proteins and globular proteins based on the shape classification. Sa may fibrous proteins, guys, they provide a mechanical and structural functions, providing to support the cell walls as well as the whole organism. Yun nga yung ating yung ating collagen. They are insoluble in water. Malamang sa malamang, they are soluble. Kung soluble sa water po ang ating collagen, malamang sa lamang, hindi na. Pagkaligo natin ay tunaw na tayo. Okay, kaya nga tayo naliligo kasi nga collagen. Kaya nga ayun, no, insoluble in water. So both internally on the surface and many hydrophobic amino acids. Polypeptide chains from the long filaments or sheets. So the interaction of polypeptide chains that forms at the fibrous protein here. So ano ba? We have the fibroin, collagen, alpha keratins, and elastin. And then next naman sa may based on shape, we have the second shape, which is the globular proteins. The globular proteins is more complex than fibrous proteins kasi nga minsan siya is made up of quaternary structures such as our heme. And they are kuha natin, nagbabago sila based on their motifs, domains, and tertiary and quaternary structures. In addition, meron din sila mga secondary structures. Okay? And they are generally soluble in water. Okay? When we say soluble in water, they tend to have a hydrophilic property. Diba? Yan yung ating mga storage proteins such as the albumin. We all know that. And then ayun, so ano ba yun? We have the enzymes, the hormones, membrane transporters and receptors, and then the IgA, and then the storage proteins, which is almond. Okay, index classification of proteins. For the third classification, we have the enzymes, biological functions. Okay, enzymes, biochemical catalysts, and transport proteins, and storage proteins. So there are three biological functions of our proteins. Enzymes natin, guys, ito yung catalyst natin sa mga living organisms natin. Increase the rate of reaction by the factor of million, guys. Ganyan siya abilis. And then merong experiment dito na kung saan yung hydrogen peroxide, if you are all familiar, lagyan mo ng hydrogen peroxidase, guys, magkaaroon agad ng evolution of carbon or hydrogen at gas doon sa may decomposition of hydrogen peroxide as compared to the non-catalyst reaction. So, hindi ko kasi siya mapapakita kasi nandun siya sa may enzyme kinetics. But I suggest na tignan ninyo yung difference, guys. Sobrang nakakamangha. Okay? And what are those? We have the kinases, mutase, and transferases. And yung mga enzymes natin are based na may suffix na ases. Ayan, kung mapapansin niyo. And there are 6 types of. Okay, saan na i-discuss. And then for the transport proteins, ito naman, they are the small molecules in the organic and inorganic. Many small molecules daw, organic and organic, are transported in the bloodstream and extracellular fluids. Yun nga, we have the anti-transport What are the example of our transport proteins? We have the hemoglobin that transport the oxygen in our body. We have the transferrin, and those are in meron din tayo mga lipoproteins natin. Okay? So, transport proteins and then enzymes. For the last, we have the storage proteins. Ang alam ko dito is the egg yolk na kung saan matatagpuan yun ang mga albumin natin. So, mga storage proteins natin, guys, matatagpuan is yung mga ferritin natin sa katawan natin that stores iron in a non-toxic form. Okay? And then we have the milk caseins that reserve of amino acids for the milk. So, casein. And then for the egg yolk phosphobiotin, it contains high amount of phosphorus and prolamins and glutenins, the storage proteins of our cereals. So there are three biological functions: catalyst, transport proteins, and storage proteins for storage. Ayan. And there are also other classification of proteins based on their biological functions, such as the hormones, contractile and motor proteins, yung mga actin and myosins natin, defense proteins, receptor proteins, structural proteins, and storage for energy. So marami pa ng functions yung mga proteins natin. And you could recall, di ba, yung nucleic acids natin na mag-central ng molecular biology na nagtatransport ng proteins natin, nagtatranslate, translation processes. So, ayun, it affects our body holistically based from that. And then, punta tayo sa may levels of protein structure. Ano-ano ba yan, sir? Okay, isa-isahin natin. We have the primary structure, secondary structure, tertiary structure, and quaternary structure. And papalaki sila ng papalaki. Okay? Isa-isahin natin to. Pero dito tayo magtatagan sa may primary structure natin, guys. Kasi after nito, yari na ang discussion natin. For the primary structure is the sequence of the amino acids in the peptide bond. Okay, since we all know that the amino acids are linked by the peptide bonds to form a polypeptide chain. The polypeptide chain has polarity. The amino end is taken to be the beginning of the polypeptide chain, diba? L stereoisomers. Okay, and it's striking fact that each protein has a unique, precisely defined amino acid sequence. So may kanya-kanya silang sequence. If kung sisipagin ka, guys, I suggest you visit the protein data bank site. Makikita nyo naman yan. And each protein, guys, has a unique amino acid sequences na na-sequence ng mga scientists natin in order for us to provide some information mga different kind of proteins natin. So protein data bank, I suggest that you search it. Okay, ayan, inulit ko lang din, peptide bonds, and magpo-form tayo ng water. And then yung natin dito, guys, yung ating peptide bond natin has a planar property. Okay, ayan. Consider this. Yung amino acids chain natin dito, we have the amino terminal end. And then the peptide bond, which is a planar, ano, NCC, what is this? That I think this is serine. And then here we have glycine, and under NCC, we have what's this? This is tyrosine, and then the alanine, and then what this is leucine. We have leucine. So ayan yung mga kon natin. Na pagsama-sama nyo na. Okay? Kuanin nyo na to Kunin lang natin dito. Okay? Kung mapapansin niyo, di ba? Ito ay pinagsama-sama. Pinagsama-sama natin to. Let's say, for example, hanggang doon lang sa may second chain kukunin ko we have the H3N, okay, naka-isoelectric point naman siya, so that will not be bother. See, we have this serine, H molecule, syempre C, double bond O, OH. This is the serine molecule. And then dito sa may NH2 natin, NH3 positive here, okay, NH2, okay, suppose, dapat ganun lang natin neutral to. Ayan, ganun natin neutral. NH2 here, and then ano yung pa natin dito? This one is glycine. I-erase ko nga. Erase natin to. Okay? Para for the convenience of all. And then here, that is N, and then H, and then H. And then we have this glycine here for the peptide bond here, and then OH. Now, kapag pinag-combine ito, guys, ito, tatanggalin ninyo yung H natin. Tatanggalin ninyo yan in order to form a water. Now, itong C na to, and then the N na to, ito yung magbabanda natin in order to form a peptide bond. So, magiging new structure na natin dito, gagawin si NH2 to NH2 here, that is C, that is CH2, COH, and then H here, then C, double bond O, nakaband na yung N sa peptide bond here. Ayan no, ito na siya guys. So, N, and then H, kasi natanggal yung H, and we have C, H, H, then C, double bond O, and then OH. Na kung iduduktong niya naman tong ating tyrosine, guys, ganun lang din yung gagawin nyo. Same mechanism. We have this N, H, H, and then C, and then we have CH2, and then C, the aromatic ring group here for the OH, the tyrosine group. Ayan. So, ganun lang yung gagawin ninyo. Tanggalin niyo yung H, pang-bond din ninyo yung C, and then N. So, ganun. Take note, guys, that it has a planar property. And then so and so forth until you meet the carboxyl end dito sa may group natin. Okay? So, ganon. And then next natin for our sample problem, ayan, this is the polypeptide structure of hormone bradykinin, which is si arginine. Ayan, guys. Tignan ninyo, it always start at the N terminus, diba? NCC, 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 and then ends with the C terminus. So start with N and end in C. So R, ito yung arginine. Then dalawang proline, mapansin niyo, we have glycine, which is G, then phenylalanine. And then what is this? This is serine, diba, guys? And then we have this proline again, phenylalanine, and then R. So ganon. Kung mapapansin ninyo, ito yung amino acid sequence natin which papasok sa may primary structure natin, guys. Ayan yung amino acid sequence ng ating hormone bradykinin. Okay? So yun lang yung point ko. Kung gusto ninyong guys, kung sisipagin na kayo, I suggest you draw the molecular structure of your name kung may papasok man. Pero pag may nawalang letters na may isang pangalan ninyo, so wala. Ayan, kaya kong i-drawing yung pangalan ko using the amino acid sequence here. Ayan. So medyo nagtitibid tayo sa time. I suggest na gawin nyo na lang ito sa may bahay ninyo. Pero may drawing on ito, guys. Okay? Ayan. For the sample problem here. Ganon. Ang gagawin nyo lang mechanism, guys, is here. Pagkasama-samahin nila yung amino acids na A, R, W, I, L, which are those the alanine, arginine, tryptophan, isoleucine, and leucine. So, ganon. Okay? Proceed na tayo. The polypeptide chains are flexible, pero sila ay conformationally restricted. Kasi nga, it has a partial double bond character and you cannot rotate it. Okay? Hindi siya ma-rotate. And planar siya. Okay? And then, most of the peptide bonds in proteins are in a trans. When you say trans, it is in the opposite side in order to para ma-lessen natin yung steric hindrance natin sa mga molecules natin that tends to like here dito. So, magkakaroon ng destabilization dito. So, mga ganun lang yun. And then the freedom rotation of two bonds of amino acids here, ito, take note, guys, na hindi ito sa may peptide banda. Nagkaaroon sila ng rotation dito, kaya tayo nagkaaroon ng mga alpha helices and beta sheets amino acids natin. Ayan, no? The freedom rotation about two bonds of each amino acids allows proteins to fold in many different ways. So, nagkakaroon sila ng folding. Pero magkakaroon din tayo ng abnormalities nun. Hindi lahat ng mga folding, guys, has a beneficial aspect. Meron din tayong mga nauuwang sakit kapag nagkaroon ng misfolding of ang mga proteins natin, such as the prion diseases, and ayun. Okay, mamaya ma-discuss ko. Ayan, so we have the steric exclusion for the primary structure. And then short polypeptide chains are sequenced with automated procedure. Meron silang automated procedure in order for that to sequence. And then the large sequence proteins must be sequenced in smaller segments. So i-divide niyo siya isa-isa para magkaroon ng. Pero may sequence naman. So ganyan, we have the level structure, the primary structure protein. Isa is take note, guys, nagkakaroon tayo ng sequencing dito. Ayan, in smaller parts. And then ayan yung mga residues natin for each polypeptide. I-sequence niyo. Ayan, isa-isa, and then wala. Meron tayong natin, mga structure. So in terms of breaking disulfide bonds, we have here, ayan, cysteine, oxidation and reduction. Magkakaroon tayo ng ganyan natin. Ito rin yung inaka natin, guys. Ang application nito sa ating biochemical engineering is yung mga pagpapakulot natin. Okay? It is a formation, it is a reduction and oxidation process in order to curl hair. Okay? And then ito guys, itong mga biological source na ito. Let's say we have a peptide, polypeptides, and then you want, if you wish, to know the sequence of that polypeptide. Ito yung mga reagents na pwede ninyong gamitin in order to determine the sequence. I-analyze nyo siya, and these are their cleavage points. Okay? Ano ba tong cleavage point natin, guys, na to? When you say cleavage point, guys, doon sila humahati. Okay? Doon sila humahati. Let's say dito, di ba, we have a common structure of amino acids na may sa may N terminus or amino group. Sorry, that's not one. Ayan. Then C double bond OH. Kapag sinabing C, ang cleavage point niya is sa may banda dito. Okay? Dito. Pero kapag ang cleavage point niya, guys, is N, ang cleavage point is banda dito. Okay, ano ba yung sinasabi ko niyan? Suppose that we have this polypeptide chain. Ito meron tayong andito. NCC, hindi ko na isasulat guys yung ko na. Isuot ko na. We have this here, we have NH2, and then H, and then we have R. We have NH here, and then R, and then H, and then C, double bond O, and so on and so forth. Kapag C, ang cleavage point niya, kunwari, suppose that is a lysine. Okay, lysine to. Kunwari, cleavage and arginine, kapag naglagay ka doon ng trypsin, ang cleavage point niya is dito, sa may parte dito. So, mahati yan, di magkakaroon ka ng dipeptide. Pero pag ang cleavage point, let's say, for example, is nasa N, and let's say this one's tyrosine, ang cleavage point niya, pero, ayun, kunwari dito na nga lang, L, R, Y, M. Ganyan, cleavage point niya is sa may gawing kanan naman. Okay? Let's say, kunwari, this tetrapeptide here, nilagyan ko ng trypsin. So, ang cleavage point niya is lysine and arginine. So, ibig sabihin, ito, we have arginine, ang cleavage point niya is sa may bandang kanan. Diyan yung cleavage point natin. Diyan yung cleavage point. Pero kapag ang nakasulat dito, guys, is N-terminus, therefore, hindi dito yung cleavage point niya. But rather, dito sa may kabila. Okay? Pero dito, sa may kabila, sa may ko natin dito, we have this pepsin. Let's say we have yung pepsin natin, naglagay ang pepsin, and then ito yung cleavage niya. Okay? And let's say ito, nag-apply tayo pepsin, meron daw siyang tyrosine. So, N-terminal. Ito yung tyrosine natin. Ito yung tyre. Hindi naman sinabing sa may C, di ba? Sinabi sa may N. So, ang cleavage point natin is sa may gawing left. Okay? Sa may gawing left. N-terminus, C-terminus. Okay? Get it? So, let's proceed to our discussion. And we also have the C-terminal analysis na kung saan yung mga gumagamit ng mga enzymes in order to cleavage our proteins here. So, we have this carboxypeptidase ABC and phosphatidase. So, ano ba application yan, guys? Consider this sample problem. Oxytocin could be sequenced directly by C-terminal analysis and a series of Edman degradations. But it provides a simple example how structure can be pieced together from fragments. Okay, dito tayo sa level 1 sample problem. Ito yung level 2 natin. Okay. And then after cleavage of the disulfide bridges, gives a mixture that includes the following peptides. Ito yun. Ito daw yung mga peptides natin. So, paano natin isi-sequence yan? Madali lang yan. Okay? Kasi nga, ayan, kuha na natin. Ang gagawin nyo lang dito, guys, in order to sequence this, is pagtatagpiin ninyo. Okay? Pagtatagpiin ninyo yung mga yan. So, simulan na natin ang pagtatagpi. Okay. Kung mapapansin ninyo, ayan, saan kaya pwedeng iduktong to between these peptides here? Kung mapapansin nyo, ni Aile ay sinulubos dito sa may pattern na to. Ayan, di ba? It contains here cis. So, sila yung magka-connecta. Ayan, let's overlap those. Okay, we have cis, tyrosine, tryptophan, I shall say, tyrosine, Aile, GLN, and ASN. And then, iduktong natin the Aile, the GLN, ASN, and then cis. Okay, now ito naman sunod natin. Saan kaya mag-overlap ito between the amino acids here? Kung mapapansin niyo, it overlaps here, it also overlaps here. Pero saan yung mag-overlap? Diba? Dito sa may gawin na to. So let's continue the structure here. Sa may kabila, itapat natin siya dito. Sa may cis. Tapat natin siya na GLN, ASN, cis, na tumutugma rin doon. And then pro. Ayan. So, paunti-unti na natin. And then dito, saan kaya natin pwedeng iduktong to? Okay? between these peptides. Kung papansin niyo, we have a pro residue here. Di ba? So dito na natin siya dodok to. Pro residue, leucine, glycine, and NH2. This is supposedly, sorry guys, ano typo ko dito. This is supposedly a C-terminus. Okay? C-terminus dapat to. Typo lang ako, sorry. And then next, saan kaya natin pwedeng idokdong to? So hahanapin natin yung patterns. Tyrosine, cysteine, tyrosine, isoleucine, glycine, ASM. Kung papansin nyo guys, ito ay papasok na siya dito sa ating fragments nito. So no need to write this. Okay? And then itong last fragments natin, saan kaya siya mag-overlap? Cysteine, cysteine, proline, leucine, and glycine. So no need to provide this kasi ito ay nag-overlap na dito sa may terminus na to. So therefore, pag tugma-tugmain mo lang yan dito, the sequence of the oxytocin guys will be cis, tyrosine, isoleucine, glutamine, asparagine, cysteine, proline, leucine, glycine, and we have the C terminus. So that's the sequence of our natin, oxytocin, in 3-letter symbol. Pero pag 1-letter symbol, that is, ano ba yung cysteine natin? I have C, Y, I, glutamine, that is Q, asparagine, that is N, C, P, L, G. So that's the sequence of our oxytocin. Level 1 sample problem. Punta tayo sa may hardcore. Okay. So bear with me. Skip natin itong part na to kasi this will be your learning activity. Ito ang kanin natin. Bear with me guys. Okay. Determine the amino acid of a sequence composition of 2 alanine, 1 arginine, 2 tyrosine, 1 valine, 1 asparagine, and 1 methionine. So, ito yung mga pan natin. These are our amino acid residue. We have dalawang ala, isang R, dalawang tyrosine, so that is dalawang Y, isang valine, isang aspartate, so that is E, and then methionine. So ito yung mga ginamit na steps daw natin. Ito daw yung mga ginamit. Okay? Sa E daw natin, partial hydrolysis yields. Ito daw. When you say a partial hydrolysis, guys, that is the N-terminus. Yun yung mga initial. Kung mapapansin ninyo, ito, what's this? That is ALA. And what's this? That is valine. So meron na tayong C-terminus. Ito yung initial natin. And then the rest, so check na to. Check na to. So the rest are A, R, Y, Y, E, and M. That is for letter A. For letter B, chymotrypsin treatment renders 2 tetrapeptides, each containing an ALA. Ano ba yung chymotrypsin natin, guys? Sige, i-paste ko rin to. Ayan. Nasaan na? Wait lang. Sige, kunin muna natin to. I-cut muna natin, guys. Sige. Okay, bear with me. Hope you understand. I'm trying to make everything convenient para sa inyo. Ayan. So, ayan. Okay? Let's continue. Sa may chymotrypsin daw, guys. Don't worry, guys. You don't have to memorize this. Mga BS Chem lang magme-memorize nito sa mga major ng mga biochemistry. So, open ito. Okay? Sa chymotrypsin daw is, meron daw tayong 2 tetrapeptides. Ano bang kiniklib niya? The lysine and arginine. Ito ba, nasa may arginine natin? May arginine. So, saan ba yung arginine natin? Nasa C-terminus. So, ibig sabihin, we have 2 tetrapeptides. We have A, V, and then this unknown amino acids, let's put it a null value. And then R, C-terminus, di ba? 2 tetrapeptides, so 4. That is 1. And then, we have this unknown sequence na Y, Y, E, M, or ano ba? Kulang ba ako ng isa? And A. Both containing alanine. Ito unknown to So any of these 5 dito. Nilagyan ko lang kasi 1. And then sa may C daw, trypsin treatment of one of the tetrapeptides, so any one of these, produce a dipeptide. So ano ba yung trypsin natin, guys? Ay, sorry guys, wait. Chymotrypsin. Ito pala. Sorry guys. Chymotrypsin pala yung sinasabi dito. Wait. Chymotrypsin. So let's X this. Okay. Sa may chymotrypsin daw, okay, nag-yield daw dito ng 2 tetrapeptides. 2 tetrapeptides. So A, that is A. Ano ba yung inuan natin? That is sa C-terminal. So AB, that is ano to? That is a null value. And this one is Y. So that is Y kasi nga, tyrosine, that's Y. And then tetrapeptides na, ano ba? We have A. So both containing alanine daw. So therefore, ang isa na to is A. But we're not sure this is the N-terminus. Okay? A, and then R, isang Y, and then E and M. Pero dalaway natin. Ibig sabihin, based from this, sa C-terminus, wala siyang ma-equilibrate dito. So therefore, the C-terminus will be Y. Kasi wala naman siyang equilibrate na kanan dito, di ba? Nagigets niyo? Oh, if you don't get it, please question. So A, so therefore, we have this A, the lagyan asterisk kasi unknown sequence, R, E, M, and then Y. So we determine the two structures. So nakaka-3-0 na tayo. So sa may C naman natin, trypsin treatment, one of the tetrapeptide, any of here, yields a dipeptide, produce 2 dipeptides. So, at trypsin natin, ang ano bang iniklib niya? D-lysine and arginine. So, dito wala naman dito. Kasi kapag dito, guys, it will produce a tripeptide and monopeptide. So, ibig sabihin, dito tayo. Sa may C natin, ibig sabihin, so it's either, let me have this, okay, null value of A, asterisk A, and then we have R, kasi di ba nag-cleave daw sa may C-terminus. And then dito, it's either E or M, and then Y. Okay? Take note guys ha, null value to ha, hindi dapat tayo sure dito. So lagyan natin bracket. Okay. And then treatment of the same tetrapeptide, ito rin daw, yields a tripeptide and trityrosine. So ano kaya nangyari doon? A tripeptide and a tyrosine. Ibig sabihin, yung R natin dito guys, the R, nag-cleave, yung cyanogen bromide natin, ito is methionine at C-terminus. So, C yung katabi niya, yielding a Y. I see. That is M. Kasi di ba methionine yung kiniklib niya sa looking anan? So, that is Y. Ibig sabihin, we can conclude guys, that this one is A. Kasi nga, a tripeptide of methionine and Y. So, our tetrapeptide here is we. Army. So, ano natitira na lang? The E. Pero, ayan, no? We can assume na dito na siya. Kasi ngayon na lang mga natitira. Pero, treatment with Sanger's reagent of the intact octopeptide followed by acid hydrogenates ASP, aspartate, which is E. Ito daw may naadoktong na E dito. So, that is AVI. So, therefore, the sequence of the amino acid of our peptides, polypeptides here are A, ito ay E na siya dito. Sa D, that is AVI. Okay, that is A, V, E, Y, and then R, me. So, nameta, ayan no, octapeptide. Ayan. So, these are the sequence, this is the sequence of our polypeptide based from the analysis and simultaneous treatment of our reagent. Okay. So, thank you guys. Okay, punta na tayo sa ating next and medyo naggagabi na. Okay. So, punta na tayo. I will make this fast na. Ayan. So, we can also synthesis peptides and proteins chemically using the Merrifield synthesis. Magkakapagpan tayo dyan. Na-discuss na sa may organic chemistry ninyo. So, these are the reaction mechanisms, but I will not discuss this thoroughly. And then here, so pupunta na tayo guys sa may secondary structure ng proteins natin. The secondary structure is the spatial arrangement of amino acid residues or its orientation in space. Di ba? Ano na yung sequence kinukulitan? So the polypeptide chains can fold into a regular structures, di ba? They have folding as discussed kanina. Nagkaroon sila ng pre-rotation. And then those are the two components, okay? Yung two major structure nun is the alpha helix and beta sheets. Okay, the alpha helix dito are stabilized by the NH and carbonyl group of the main chain. So pag ganito siya, it can be right-handed or left-handed chain. Meron siyang translation of 1.5 angstrom and 3.6 amino acids per turn, or it has a measurement of 5.4 angstroms per turn. So pag ganyan siya, pa-helix yung secondary structure natin. Then for the natin, ayan, meron tayong right-handed and left-handed. So how can you determine here? By using our hand here. Ayan. Merong articles doon. I think it is here to determine the quaternary. Ayan. If you wish to determine the structure of our amino acids, if it's either left-handed or right-handed, you may follow this article. Yun lang, hindi ko makita. So, ikaw na natin muna. Ayan. And the next natin here, ayan, alpha helices, right-handed, left-handed. And then the beta platelet sheet are stabilized by the hydrogen bonding between the polypeptide strands. So the distance between chicken atoms are 3.5 angstrom. Mamaya makikita natin ang applications niya. So it can run in a opposite direction or in the same direction. Anti-parallel and parallel beta sheet. And we have the Turnison loops, the hairpins and turns, and gametes. So ano ba yung special type of alpha helix natin in beta sheets? Sa may alpha helices natin, we have the alpha ertin, collagen, and kapag sa beta sheets, we have 6-fibroin. Sa may alpha-erotin, we all familiar, that was discussed earlier na it is the primary component of wool and hair. It is a right-handed helices, two right-handed daw. Ganun yung helices niya. And a coiled coil, left-handed. So coiled na-coiled na nga, kinoil pa. So ganun ka-twist po ang ating mga alpha-erotin. And it has a 1,000 angstrom and contains heptad repetition. Okay. And then for the collagen, it is also a alpha helix, the most abundant protein in mammals. Kasi nga, it is made up of our skin. So it's the most abundant protein sa mammals. Ang fibrous component na guys ay nakikita sa may skin, bones, tendons, cartilage, and teeth. Kaya siya sinabing most abundant proteins in the mammals. Pwede siyang rod-shaped molecule and it has a repetition of lysine, proline, and hydroxyproline. Yan yung repetition residue ng ating collagen. And then the overall, kapag binagsama ninyo 3D structure, doon na magkakaroon ng 3D secondary structure. Pag binagsama sa secondary structure, doon na magkakaroon ng 3D structure. Na kung saan guys, kung sa may secondary structure, ang intermolecular forces na involved doon is yung ating hydrogen bonding. Pero dito, there are 4. And what are those? The hydrogen bonding, again, the hydrophobic interactions within the non-polar aliphatic group in the polypeptide backbone, the disulfide bridges, and ionic bonding. So there are 4. Hydrogen bonding, hydrophobic interactions, disulfide bridge, and ionic bonding. So it has some motifs because it is the combination of secondary structure and super secondary structure. So ito yung mga structures nila ng proteins natin. And makita niyo yan, makita dyan sa mga protein data bank natin. Okay? So ayan, for the quaternary structure is the structure of the natin, mga pinagsamang tertiary structure. We have the hemoglobin, pinagsamang, ayan, dalawang alpha and dalawang beta. So ayan, so primary, secondary, tertiary, and quaternary structure of proteins natin. And then we have yon, pagka as discussed earlier, kapag nagkaroon ng misfolding sa proteins natin, magkakaroon tayo ng mga diseases. Such as dito sa may pinapakita na the bovine spongiform encephalopathy or mad cow diseases, and the Creutzfeldt-Jakob disease in humans, Skripinship and Prions diseases, na mga neurodegenerative diseases natin, guys, na pwede magpa. And then, ito yun din, yung sickle cell anemia natin, na kung saan nagbago lang yung sa may natin, sa may nucleic acids, is nagbago lang yung isang arrangement ng isang base or mga codons natin is nagkaroon na ng sickle cell anemia natin. So those are ma-encounter natin kapag nagkaroon tayo ng misfolding or mabago yung sequence ng ating mga amino acid sequence in the proteins natin. So napaka-crucial nun. Kaya genetics pa rin talaga mag-play ng role kung magkakasait tayo or hindi based dun sa may chromosomes natin. Okay, and I think that ends our discussion guys for our proteins. And I hope you learned something guys. So see you in our next lecture. And good luck, guys. Thank you for listening. And my name is Arvin Atanyan, your instructor. Thank you very much.